In this chapter, we're talking about how we use the microscope and how we interpret the image. So there's a lot of skill level and technique, the right tool for the right job, etc. In the pictures on this slide, we are seeing a bright field microscopy on the left, rendering a darker image on a lighter background. And then on the right, we're seeing a dark field microscopy. Increases the contrast, rendering a brighter image on darker backgrounds. And that's just a little bit of a peek into what we'll learn about in this chapter. Before we can understand microscopes, we need to understand light, because microscopes depend entirely on how light behaves. If light isn't interacting with the specimen properly, we won't get a clear or accurate image, no matter how expensive the microscope is. And I looked into this. You can get a microscope for under $30 all the way up to over $1 million when we're talking about those industrial strength electron microscopes. Light behaves in predictable ways. Now, they're predictable ways because we have scientists who have studied light and the movement of, the waves of, all of the science behind that. So now we can predict how light should move. And those behaviors determine what we see through a microscope. In microbiology, we're not just looking, we're controlling light to create contrast between the specimen and the background that specimen is on. If you look at figure 2.2, in the book it's 2.2, the amplitude is the height of the wave, whereas the wavelength is the distance between one peak to the next. These waves have different frequencies, as you can see on the picture on the right, or the image on the right, or they have like different rates of vibration. The wave at the top has the lowest frequency, since it has the fewest peaks per unit of time, and the wave at the bottom has the highest frequency. So these terms, reflection, absorption, interference, and diffraction, describe how light interacts with objects. So for example, you have absorption, which helps create contrast. You have diffraction, and diffraction that can actually limit how clearly we can see very small objects, and that's why there's limits to what a light microscope can resolve. In this figure, we can see a petri dish, which is either plastic or glass, so it's transparent, you can see through it, is transmitted through that material. Whereas material like this slice of iron meteorite is opaque. It has opacity. We can't really see through it. So it's very difficult to study the image, the material. Refraction is the bending of light as it passes through materials. So you can imagine, depending on the material, it's going to bend differently. Microscope lenses are designed very precisely to bend light in a way that brings the image into focus and magnifies it. Even small changes in how the light bends can affect the image clarity. And that takes, some very well-made microscopes, but also the skill level of the person using it and how they are setting the light intensities, etc. One figure here we have refraction occurs when light passes from one medium, such as air, to another, such as glass, changing the direction of the light rays. In B, as shown in this diagram, light rays passing from one medium to another may be refracted or reflected. The other figure here is a straight pole that appears to bend at an angle as it enters the water. This optical illusion is due to a large difference between the refractive indices of air and water. And you can try that at home when you put a straw or a pencil or something in your glass of water. And you can see how that it's kind of an optical illusion that it's not straight anymore, that it bends a little bit or shifts. Focal point and focal length determine how clearly an image is focused. So you may have struggled with getting something into focus, and you were adjusting the focal length to bring the image to that focal point. So that's what you're trying to do, adjust the focal length to bring the image into focal point. If you look at letter A, a lens is like a collection of prisms. And then B, when light passes through a convex lens, it is refracted toward a focal point on the other side of the lens. The focal length is the distance to the focal point. And then letter C, light passing through a concave lens is refracted away from the focal point in front of the lens. So visible light is just a small part of the electromagnetic spectrum. Some microscopes, like electron microscopes, use completely different types of energy, which is why they can't achieve much higher resolution. We have the electromagnetic spectrum that ranges from high frequency gamma rays to low frequency waves. Visible light is a relatively small range of electromagnetic frequencies that can be sensed by the human eye. On the electromagnetic spectrum, visible light falls between ultraviolet and infrared light. So that's that bottom picture. The top picture, the fluorescent dyes absorbed by these bovine pulmonary artery endothelial cells emit brilliant colors when excited by this ultraviolet light under that fluorescent microscope. So now we're going to shift to microscopy, how humans first began seeing microorganisms. What a huge turning point in science when suddenly this entire world of organisms that we couldn't see before, that were always there, are suddenly visible. So here we have the real beginnings of microbiology as a field. So if you look at the picture on the right, we have Robert Hooke's compound microscope, and he used it to view many things under it, but the most well-known one is cork cells, which cork is from a tree, and it's really cool. You should look up how they harvest cork. It's so cool. Those are plants. So looking at plant cells under the microscope, they kind of look like cells that monks lived in, so he called them cells. And both of the engravings right here on this slide are from his work Micrographia, which was published in 1665. Then you move on to Antony van Leeuwenhoek, living from 1632 to 1723. But he's credited with being the first person to observe microbes. So not just looking at things closer up like Robert Hooke, but he saw things moving, and he called them animalcules, these prettily swimming about, wee little beasties. And those were bacteria and protists that he was seeing. And he looked at things like vomit and semen and diarrhea, anything he could get his hands on. He was just looking at everything under a microscope. He was the one that, worked with clothes and threads. So he looked at a lot of that underneath too. We talk about thread count when we talk about sheets. So maybe he was concerned about the quality of his craftsmanship. His microscopes were very simple. As you can see in that middle picture, that's a replica. But they were more powerful and provided better resolution than the compound microscope of that day. So it's kind of a little bit better than what Robert Hooke was using. Instruments of microscopy. We're going to look at the tools we use today. Different microscopes are designed for different purposes, so there's actually no one perfect microscope that does it all for us. The light microscope. So this is the most common type of microscope. Total magnification equals objective lens times the ocular lens. So here's something very important. Magnification is not the same as resolution. Resolution is your ability to distinguish two objects as separate, like that clarity. You can magnify something a lot, but if the resolution is poor, it's going to look blurry. So staining is something we use to improve that contrast so we can actually see the specimen. So let's take a look at this picture here. If you look at the very top of the microscope, that is your oculars. So sometimes there'll be one or two. So biocular or monocular. If you are using one with one ocular, you're not supposed to close the other eye while you're looking through. It's pretty difficult to train yourself to do that if you've always kind of closed one eye looking through. But here we have two, and that is usually 10 times. So it's magnifying something 10 times. And then you have the objective lenses, and you see different ones in that picture. So on the microscopes that we work with in the college classroom, we have a 4 times, a 10 times, a 40 times, and 100 times as the objectives. So if I want to put the shortest one on, that's the 4 times. So it's 4 times the objective lens, 4 times, and then the ocular lens, I told you it was 10 times. So 4 times 10 is 40. So you're seeing something 40 times closer. Then you focus in on that. It's best to focus in on something at the lowest objective, and then you move on to the next one, and you go from there. So we'll have some labs that help you learn the microscope. Oil immersion improves resolution by reducing light bending. So when you do the 4 times objective, then the 10 times, so that would be 10 times 10, that's 100. Then you have the 40 times, that would be 40 times 10, that's 400. Now, light microscopes will have the 100 times objective. So 100 times that 10 times ocular. Now we're getting into 1,000 times magnified. So the oil immersion helps to reduce the light bending. That's the way we use this particular objective. So normally light scatters when it passes through air. The oil has a similar refractive index to glass, so it keeps light more focused. This is essential when viewing very small organisms like bacteria. And then here in the picture, oil immersion lenses like this one are used to improve resolution. And then because immersion oil and glass have very similar refractive indices, there is a minimal amount of refraction before the light reaches the lens. Without immersion oil, light scatters as it passes through the air above the slide, degrading resolution of the image. Basically, that's a restatement of what I just said, but just to drive home the point, I guess. Phase contrast microscopy. So this enhances contrast without stains by manipulating light waves. It takes advantage of differences in how the light travels through different parts of a cell. It's going to allow us to see internal structures in living cells. Remember, that's why we don't want to stain them. We want to keep them alive. So the figure here compares a bright field image on the left with a phase contrast image on the right of the same unstained simple squamous epithelial cells. The cells are in the center and bottom right of each photograph. The irregular item above the cells is just acellular debris, just stuff there. Notice that the unstained cells in the bright field image are almost invisible against the background, whereas the cells in the phase contrast image appear to glow against the background, revealing far more detail. So you can see a lot more detail there. And the diagram of the phase contrast microscope illustrates phase differences between light passing through the object and the background. These differences are produced by passing the rays through the different parts of the faceplate. The light rays are superimposed in the image plane, producing contrast due to their interference. Now, don't you worry, I'm never going to get this specific on an exam, but it is interesting to see how each one of them, just knowing the right microscope for the right job is important. This differential interference contrast, it's going to produce this 3D-like image. It uses 2 beams of light and creates contrast based on differences in thickness and density. So it's more of a detailed view of these structures. It's actually pretty cool looking. And then this image here, I'm afraid to try to pronounce it, but it is a fungus, and it's actually a chronic skin infection common in tropical and subtropical climates. So that's kind of cool. It causes chromoblastomycosis. So here we have fluorescence microscopy. It uses fluorescent dyes. I mean, that makes sense, right? That emit light when excited by a specific wavelength. And this is widely used in medical diagnostics, research, identifying specific pathogens. So those are cancer-causing organisms. You can even label specific molecules inside cells. The picture here, we have a direct immunofluorescent stain It's used to visualize this bacterium that causes gonorrhea on the left there. And then an indirect immunofluorescence stain is used to visualize this larvae. It's a parasitic worm that causes, it causes schistosomiasis, schistosomiasis, an intestinal disease common in the tropics. Well, I hope I don't get that. And then letter C. So that's another direct. So the first one was direct immunofluorescence stain. The second one was indirect immunofluorescence stain. And now I'm talking again about direct immunofluorescence, a stain absorbed by primary antibody, which binds to the antigen. And then the indirect one, it is absorbed by a secondary antibody, which binds to a primary antibody that has bound to the antigen. So as you can see in their picture, just one more level to that one. Confocal microscopy uses lasers to scan thin layers of a specimen. These layers are combined to create a 3D image. So this is going to be really useful for thicker samples, that it can kind of take it apart and then reassemble it in the image. Views structures such as this roof-dwelling cyanobacteria biofilm. The electron microscope, expensive. They use electrons instead of light, so that allows for much, much higher resolution in those really cool pictures. Because electrons have shorter wavelengths than visible light, they can reveal much smaller structures. Okay, let's take a look at the differences between the TEM versus the SEM. So we have the transmission electron microscope. It shows internal structures. It requires very thin samples, and it produces a 2D image, whereas the scanning electron microscope shows surface structures and produces a 3D image. So it really depends on what you're trying to study. Are you trying to study the inside or the outside of these organisms, cells, etc.? The illustrations there are, they compare the components of both of those microscopes. So you can take a look, but you do not have to memorize that. Let's take a look at this lovely image. We have the transmission electron microscope image on the left that shows a biofilm, very well-defined internal structures of the cells because of the varying levels of opacity in that specimen. And then you have B, that's going to be the scanning electron microscope image, which is color enhanced. And it is the bacterium Staphylococcus aureus. And then that just illustrates the ability of scanning electron microscope to render this three-dimensional image of the surface structure of cells. Scanning probe microscopy. These are going to allow us to observe surfaces at the atomic level. So instead of using light or electrons, it uses a tiny probe that moves across the surface of a specimen. There are two types, scanning tunneling microscope and the atomic force microscope. If you look at letter A, this STM image of a pure gold surface shows individual atoms of gold arranged in columns. That is wild. Then this AFM image shows long strand-like molecules of nanocellulose, a laboratory-created substance derived from plant fibers. Okay, here we have a great table that summarizes all the various microscopes. So every one of them has its strengths and its limitations. So the key skill in microbiology is actually choosing the right tool for the question that you're asking. Are you asking about a pathogen if someone's sick? Are we just asking to study a certain type of microorganism that's been unearthed via permafrost melting and climate change? So what are we studying? What are we trying to see? I'm just here to tell you there's more summary about microscopes and their limitations and advantages. I just didn't want you to be sad without hearing my voice on every single slide. Guess who's back? Just to say, this is more microscope summary for you. Now we're moving on to staining. So how can we make those cells even easier to see? Most microorganisms are pretty much transparent, so without staining, they are really difficult to observe and study. So we have to prepare the specimens before we can observe them. There are two key methods of preparing them: wet mounts and fixed samples. So wet mount, you're in the classroom, you have a drop of water, you have your specimen in that little drop. These are live cells. You're going to allow for observation of movement. Now, you might stain them, but, some stains will kill them. So it just depends on what you're doing with those specimens. And then you have fixed samples, which are the cells are killed and attached to the slide. They're going to be better for staining because the stain typically kills the organism anyway, and for long-term observations. So these are slides, that you have in your classrooms, prepared slides, they're called. So the figure here, so A is a specimen can be heat fixed by using a slide warmer like this one in the picture, or a Bunsen burner is used sometimes, but it's heat fixed, so it kind of sticks on there because of the heat. Another method for heat fixing a specimen is to hold a slide with a smear over a micro incinerator, and that's B. And then C, a tissue sample is being fixed in a solution of formalin. So it's a type of formaldehyde. By the way, both those chemicals, they're considered carcinogens, which is cancer causing. So I've been exposed to a lot of that when I was in my zoology classes throughout college. Worries me a little bit, but in schools, to limit the exposure, let's say you're a teacher, to limit the exposure to your students to these chemicals, close things properly, ventilate properly, follow all the directions. Okay, enough of that. And then you have chemical fixation. It kills microorganisms in the specimen, stopping degradation of the tissues and preserving their structures so they can be examined later under the microscope. So that's what formaldehyde does. It preserves, and it does its job very well. Just another side note, because I know you want to know, if you have something with a scent in it, let's say air fresheners at home, or detergents, anything with scent that says, oh, it lasts a long time, it lasts weeks or months, it's a good chance that it does have formaldehyde in it. I don't mean to bring you bad news, but I teach environmental biology, so these are the kind of things I teach in that class. But look it up and try to use air fresheners and scents that don't have formaldehyde. Okay, we prepared, and now we are staining. Stains interact with the cell structures differently based on charge. Remember, cells have charges in them. Basic dyes bind to negatively charged cell surfaces. Acidic dyes will stain the background, and that's going to create that contrast so we can distinguish the cells. In A, this image up here, the Bacillus anthracis cells have absorbed crystal violet, a basic positive stain. And then in B, the specimen Spinochloricus, a microscopic marine organism, has been stained with rose bengal, a positive acidic stain. In the last one, Bacillus megaterium, appeared to be white because they have not absorbed the negative red stain applied to the slide. So you're basically, overall, you're looking at stains with different charges and cell surfaces with various charges and working with that to create contrast. The Graham stain. This is named after a person with the last name Graham. He came up with this staining procedure to differentiate between gram-positive bacteria and gram-negative. Now, a lot of bacteria fall into one of those two categories because of the way their cell walls are built. There are others not in the gram staining categories, but when you're sick, you go in, they take a culture, they're going to run this gram stain test to get an idea if that pathogen is either gram positive or gram negative, because it matters how they treat it. Various medicines work in different ways depending on that cell wall. So you have these four main steps.
The Gram stain is named after its inventor, Hans Christian Gram. As a Danish botanist and teacher of pharmacology, he devised this important method of differentiating bacteria in 1884 that is still in common usage today. The Gram stain is used to distinguish between gram-positive and gram-negative cells. It is the most commonly used method to observe bacteria. The Gram stain provides two important features: the Gram reaction, or color of the cell, and the cell morphology, the shape of the cell. Bacterial cells can be spherical, rod, or corkscrew in shape, and nearly all clinically significant bacteria can be differentiated into these three groups. Knowing which type is causing an infection is critical to patient care. The Gram stain differentiates cells based upon differences in the structure of the cell wall. The cell wall is made up of peptidoglycan, which consists of sugars and amino acids that form a layer around the plasma membrane of most bacteria. Bacteria that stain dark blue-purple are called gram-positive. They have a thick peptidoglycan layer that retains the primary dye. Conversely, bacteria that stain pink to red are called gram-negative. They have a thin peptidoglycan layer that loses the primary dye when flooded with alcohol. The Gram stain begins by adding 1 drop of sterile saline or deionized water onto the center of the slide. Then take a sterile loop and touch the edge of an isolated colony on the Petri plate. Place the loop into the drop on the slide and swirl it around to distribute the cells evenly, so the cells are 1 layer thick. Using cells from a single colony is important, as this should represent only one cell type rather than a mixture of cells. Ideally, the culture should be between 18 and 24 hours old, because older cells may show variable results. Alternatively, you can use a collection swab from a clinical specimen or environmental sample. Do not heat the slide. Wait for the sample to air dry. Bacterial cells are then fixed to the slide using methanol. Fixing cells is important, as it prevents the cells from washing off during the staining process. Methanol fixation is recommended rather than heat fixation, as methanol preserves the cell's morphology. Heating the slide will cause cell distortion, could increase cell debris, and may cause erroneous Gram reactions. Place or hold the slide over a paper towel and flood the slide with absolute methanol for 2 minutes. Alternatively, you may dip the slide into a Copeland jar filled with methanol. Once 2 minutes have passed, tilt the slide and drain off the excess methanol and let the slide air dry. Do not wipe or blot the slide, as this can remove cells. Do not heat the slide. Cover the slide with the primary stain, crystal violet, for 1 minute. Crystal violet is a dark blue to purple dye that stains all cells. Rinse the slide with deionized or tap water to remove excess crystal violet. Flood the slide with Gram's iodine for 1 minute. The negatively charged iodine molecules act as a mordant and link to the positively charged crystal violet. This traps the crystal violet in the peptidoglycan layer. Once 1 minute is up, rinse off the Gram's iodine with deionized or tap water. Tilt the slide at an angle over the sink or tray and add the decolorizer dropwise until the violet color stops running. When the solution runs clear, immediately rinse the slide with deionized or tap water to neutralize the decolorizer. The decolorizer is composed of a ratio of alcohol to acetone. The ratio is normally a personal preference, with faster decolorizers containing higher concentrations of acetone and slower ones containing higher concentrations of alcohol. When decolorizer is added to the smear, lipids are extracted from the cell wall of Gram-negative bacteria. Lipid extraction causes an increase in cell wall permeability, and the cell becomes leaky, resulting in the loss of the purple dye mordant complex from the thin peptidoglycan layer. In contrast, the effect of decolorizer on Gram-positive bacteria is dehydration. This decreases the cell wall permeability, causing Gram-positive cells to retain the crystal violet Gram's iodine complex. Note that a thick smear will require more decolorization than a thin smear. Be sure to stop the decolorization process immediately after the blue dye stops running off the slide. Beware. Over-decolorization can result in false Gram-negative results. Beware. Under-decolorization can result in false Gram-positive results. In essence, Gram-positive cells will be blue-purple, and Gram-negative cells will be colorless after decolorization. The final step utilizes a counterstain of a different color to differentiate gram-negative from gram-positive cells. Cover the slide with safranin or carbylfuchsin for 1 minute. The gram-negative bacteria will absorb this dye, causing them to stain pink to red. There will be no effect to the gram-positive cells since they retain the initial crystal violet stain. Rinse the slide with deionized or tap water. Do not wash the slide excessively. Allow the slide to air dry by tilting it onto a paper towel or over a sink and allowing it to air dry. Alternatively, gently dry the slide by blotting it using a lint-free bibulous paper. Do not use a wiping motion, for this can remove the smear. To summarize, crystal violet exposure, 30 to 60 seconds. Iodine exposure, 30 to 60 seconds. Acetone alcohol decolorizer, only enough time to rinse away the blue stain. Safranin exposure, 30 to 60 seconds. The slide can now be viewed under the microscope. First, focus on the image using the high dry objective lens marked 40x. Then, without removing the slide, switch to the high power oil immersion objective lens that is marked 100x. This will result in an overall magnification of 1000x. Here you see two organisms routinely used for quality inspection at Hardy Diagnostics. The purple gram-positive cocci are Staph aureus. The pink gram-negative rods are E. coli. Hardy Diagnostics offers an improved formulation of gram stain called Gram Stain Advanced. This kit produces superior stain results with bright, vivid colors, even for bacteria that are difficult to stain. To test the quality of your stain and staining technique, Hardy Diagnostics offers quality control slides, the Q-Slide Gram, that are pre-inoculated and methanol fixed with gram-negative and positive bacteria. Thank you for watching this gram stain demonstration.
Let's take a look at some gram stain examples. That first picture on the left, we have gram-positive, so they're that like deeper purple. That is a staphylococci, so the COCCI, cocci, shape is that round shape. This next picture, we have a mix of gram-negative and gram-positive. So we have the gram-positive cocci, which are purple and round, and then we have the gram-negative rods. So those are those pink rod shapes, so bacilli shape. And then this last one is a gram-positive rods found among white blood cells. So these differences are so clinically important to figuring out which pathogens somebody may have. And that's like your initial treatment decisions are made off of this gram stain. So that's why when you leave microbiology class, everyone needs to know how to do that. So watch those videos for sure. Acid-fast stains. They're used for bacteria with waxy walls, so such as Mycobacterium. These cells resist typical staining, so this special technique is needed. So think about wax, right? Candle wax, wax on your car. It's just harder to get through. It's going to be very important in diseases like tuberculosis, which is pictured here. Capsule staining. This highlights the very protective outer layer called capsules. Not all bacteria have this, but when they do, they are sometimes very difficult to kill. So they help bacteria. They help bacteria avoid the immune system, and it increases their virulence, meaning that they can do some harm. India ink was used to stain the background around these cells of yeast in this first picture here, Cryptococcus neoformans. The halos surrounding the cells are the polysaccharide capsules. So polysaccharide, poly means several, saccharide, it's like sugars. So the basically it's made out of carbohydrates. And then B, you see crystal violet and copper sulfate dyes that cannot penetrate the encapsulated bacillus cells in this negatively stained sample. Encapsulated cells appear to have a light blue halo. Endo, think of endo being like inside. And if you ever see the term exo, that means outside. But these are endospores, so they're going to be inside. These allow bacteria to survive extreme conditions. It's important for organisms like Clostridium difficile. Now that one's called C. diff, in hospital settings or nursing home settings. I've had nursing students or, students who work in medical buildings and things. They have told me that this is one of the worst to treat and to clean up after. So beware if you're in that field. So this figure, a stained preparation of Bacillus subtilis showing endosporins as green and the vegetative cells as pink. Spore stain procedure. This video uses B. ligoformis, a spore-forming bacterium. Spores form after 48 hours of growth. Set up a hot water bath. Air dry and heat fix a smear of bacteria. Cover the smear with a small piece of paper towel. Saturate the paper towel with malachite green. Steam the slide for 5 minutes by placing it over a beaker with boiling water. Add additional stain as the stain boils off. Allow the slide to cool. Remove the paper towel piece and rinse with water for 30 seconds. Counterstain with safranin for 20 seconds. Rinse lightly to remove the safranin. Blot dry and view the slide in immersion oil under the microscope. Spores stain green while vegetative cells stain red or pink. Magnification 1,000 times. Spores can survive difficult environmental conditions like extreme heat, dehydration, low nutrients, radiation, etc. Magnification 1,500 times. There's even a technique to stain flagella so we can visualize it better. Those are structures used for movement. We're pretty familiar with the flagellum on sperm cells, and they're very thin and normally hard to see. And here we have Bacillus cereus, a common cause of foodborne illness, and this reveals that some cells have numerous flagella.
The flagella stain is used to determine whether a bacteria sample contains flagella, a characteristic that can help in the identification of an unknown culture.
Things you will need: Bunsen burner, inoculation loop, specimen, distilled water, microscope slides, clothespin, staining rack, flagella stain, immersion oil, and microscope.
Place a drop of water on a microscope slide approximately 1 centimeter from the edge. Next, heat and cool your inoculation loop. Then, obtain a sample from an isolated colony of your specimen. Lightly touch the sample to the drop of water without touching the slide. Do not mix. Tilt the slide at a slight angle to allow the drop to flow to the opposite end of the slide. Place the slide on the staining rack and allow the slide to air dry at room temperature. Do not heat. Holding the flagella stain dispenser upright and with the tip pointing away from you, squeeze gently to crush the glass inside the dispenser. Then, flood the slide with the flagella stain. Allow the stain to remain on the slide for approximately 4 minutes. Carefully rinse off the stain with water. Do not tip the slide as flagella are very fragile. Then, gently tilt the slide to allow excess water to run off. Allow the slide to air dry at room temperature. Add a drop of immersion oil and observe using the 100x lens of the microscope. An example of an organism positive for this stain is E. coli.
Here is our staining summary. Simple stains. One dye, basic shape or structure we're looking for. Differential stains, multiple dyes, and provide more detailed information. The gram stain is the most important differential stain. Again, simple stains versus the differential stains. Preparing for electron microscopy. So we're back talking about the TEM. It's going to require extensive preparation. You're going to have to dehydrate. So take the water out of cells. Very thin sectioning and heavy metal staining. And that's going to allow the electrons to pass through the specimen. That's an image of a microtome used to prepare specimens for TEM. B, a technician uses ultramicrotome to slice a specimen into thin sections. Samples are coated with metal like gold. That's preparing for the scanning electron microscope. So this allows electrons to interact with the surface and produce those detailed 3D images. Hope you enjoyed all this information and you made it to the end.